Yeah. Okay. Good afternoon, everybody. I think most of the iGEM teams have now arrived, and it's really good to see so many people here. And I'd like to welcome you all to the Biochemistry department in Oxford. I'm Judy Armitage. I'm a professor of microbial biochemistry, but also head of the Systems Biology Centre and one of the PIs on the Synthetic Biology CDT. And for the first time this year, Oxford has put together an iGEM team. I have to say, it really shows that iGEM teams need to come from the students together being inspired. I tried a few years ago and discovered that you can't force a group of undergraduates together to actually put together an iGEM team. They've got to want to actually come together as an iGEM team. I'm afraid I'm not going to be able to stay for terribly long, so I'm going to hand everything over to Isabel. I'm the academic in theory in charge of our iGEM team, but actually it's George Waddams who is in fact in charge, and he will be around for the rest of the day. I unfortunately have to go off and be somewhere else, but it's a really great pleasure to have here in the UK to talk to the iGEM team, Randy Rettberg, who's going to tell you about, I think, all sorts of things. I mean, all sorts of things. He's just going to, talk to you about his experiences and how this all evolved. And then we'll have more people telling you about putting together iGEM teams and what's sort of expected of iGEM teams. But again, I'd just like to welcome you all here to the Oxford Biochemistry department and hand over to capable colleague.
Thank you. Well, good afternoon again, everyone. I'm very happy to see so many of you here today. And on behalf of SYNOPS, the Oxford Society for Synthetic Biology, and the first ever Oxford iGEM team, I would wish you a warm welcome to Oxford and this wonderful event, SYNOPS. My name is Laura. I'm the president of SYNOPS, and I will be the host today for the 3 talks we have planned. Jumping right in, we will start off with our 3 talks. Then this will be followed by a Q&A panel at the end of our talk. So please keep your questions with you and try to remember them until the end of the talks. After the Q&A session, we will head down to the atrium of the building where drinks, nibbles, posters, and hopefully stimulating conversation will be for you. So without further ado, I would like to welcome our first speaker. He showed interest and potential regarding educational matters early on in his scientific career. Yes, he did. And while finishing biology in Poland, he also co-founded and directed the Center for Innovative Bioscience Education. Somehow he must have managed to fit more hours into a day, because besides this, he also set up the Bioeducation Foundation aimed at popularizing science in the same country. Currently, he resides at the National Center for Biotechnology Education at the University of Reading. Here he has been successful in establishing synthetic biology teaching kits called the UniGEMS project for the undergrad level. This also led to the formation of this year's iGEM team, which are with us here today. And now I would like to introduce him to you, so please give a warm welcome to Dr. Jarek.
Thank you very much for having me here. And I think the order of the talks is really cool because I'm just going to start at the beginning, as a beginner myself. Then we go to the firmly established in the synthetic biology scene, Richard, and finally to Nancy. So I will start a little bit louder. Oh, wow, okay. So I'll tell you 2 words about what I did before I started to do this. I was an evolutionary geneticist 2 years and 3 months ago, working in Germany on these wild mice in France and Germany. And then I decided to write a grant that required me to switch career focus and concentrate a little bit on teaching, because I like really teaching. I really love teaching, and I want to do some more of it. So I wrote the application, it failed, I wrote it again, and it succeeded. And I was doing until end of May the UniGEMS project, which aimed to develop experimental kits to facilitate teaching of synthetic biology. Our goal was to figure out a set of parts and a set of protocols that would allow any university like ours, like Rettberg, where there was no synthetic biology before, their staff to pick it up and start teaching and giving some synthetic biology experience to their students. So getting over this extra big first hoop, that was the goal. So this project is almost finished, and there will be some PDFs and protocols and parts names and sequences available online pretty soon. And it's been done there in National Center for Biotechnology Education, which is a part of the University of Reading, which develops and sells experimental kits to teach biology in high school, so 16 to 18-year-olds, A-levels in the UK. So they had all these different stuff. If you had done any practical work in biology while in your A-levels, it's probably been developed by MCBE. So since I came to, we developed this, a bacterial transformation kit that does not require pipette or competent cells are stored at -80 or -20. And we based a couple of other protocols that came out of it on this initial idea. Right. So you want to start your synthetic biology at the university that had absolutely no synthetic biology. You're a molecular biologist with expertise in something else. So you come to a place, you have this vague status as a postdoc / members of staff, so no one knows you, but you still want to do it. You want to teach students synthetic biology. So you go, you call up a meeting with other fellow members of staff, and this is the quotes of shame wall that you get from some of them. All right. So people either do not know what synthetic biology is, even microbiologists don't know what synthetic biology is. There are very high and strong walls between departments. So it's very hard to talk or to have a conversation between biologists and engineers at your university, and you somehow want them to collaborate on developing this new teaching resource. Okay? So the answer to this is this nice quote by Robert Heinlein: everything is theoretically impossible until it's done, and you should just ignore people who tell you that it can't be done. Okay? And being this outsider who came from somewhere else and is not really well established in the community, actually was a good thing for me, because no one really cares what you're doing, so you can do a couple of things that other people would not think of. And so I did. So first of all, you see how other people who are very good at what they're doing do this. You probably know Jim Hasselhoff from Cambridge, who has set up a very, very high-quality, high-profile synthetic biology program in plants. And he's been running sort of an iGEM bootcamp for his iGEMers every year for 2 weeks. They would come every day, they learn the basic techniques, they do some Arduino engineering, transform bacteria, and so on. So I went to this course, hanging out with them for a couple of days, and just sort of had the feeling how these materials that they use and they gave to the students, how they work. And of course, they are very successful team. They won in 2009 and set up a career of basic input. All right, the next thing, and this is a very important thing, you have to find a friendly engineer if you're a biologist like me. Or if you're an engineer, you have to find a friendly biologist. Because, in my opinion, this is the only way of doing proper iGEM or proper synthetic biology community at your place is with engineers and biologists talk to each other. These are very different worlds and very different languages that you have to overcome. But if you find a guy who is interested in synthetic biology and is an engineer, it makes everything much easier. And I'm going to single out Jarek from cybernetics in Reading, and his postdoc, PhD student, Yannis Zulias, who actually came to talk to me when they heard about the course I was running. And they were interested, and they went to the course themselves, and they stayed. So not only they wanted to do synthetic biology with biologists, they also wanted to have it sort of spread across the university. And Slavik being a full professor was sort of much higher in hierarchy than I am. I think his position allowed many things to happen. So you have to have someone who would let you bridge the gap between engineers and biologists that is there. All right. So then, what is helpful is to do your training or teaching synthetic biology or developing synthetic biology anyway. You don't really need to start an iGEM team right away, although that's a good way as well. But it wasn't possible for us back then. So what we did, we ran a workshop for undergraduates, a voluntary workshop. The undergraduates volunteered to come to our workshop. It was in June after all the exams were finished. So they gave us 2 weeks of their time where we did stuff, synthetic biology stuff with them. Transformation of bacteria, Gibson assembly, simulations of bacterial circuits, and so on. Thanks to connections to Cambridge, we had a connection with Andrew Phillips, a Microsoft researcher, to simulate cells in a computer. And he came to give us the talk and to give us the run of the software to simulate cells. And all in all, it was a very successful workshop. We initially had 20 places announced, and there were 60 people coming, so we had to run it twice, and still turned a few people away. And this is just an excerpt that gives you an overview of sorts of students that would come to a workshop like this. This massive field like I just went to a couple of members of staff and asked for 5 minutes at the end of the lecture during the school year. I gave 10 minutes in microbiology, I got 40 minutes at bioinformatics lecture, and so on. And I just took, okay guys, this is synthetic biology. I'm running a course in the mid-June. Write me an email if you want, or go to this website, and that's how it started. So we had students from zoology, theater arts, systems engineering, pharmacy, nutrition, food science, microbiology, management, cybernetics, chemistry, biomedical sciences, biochemistry, and artificial intelligence. So you get very, very nice diversity of people, and they can contribute various ideas and points of view about stuff you're doing that you wouldn't get otherwise. If you just announce a course during the microbiology module, you wouldn't get this sort of diversity of students. So that was great. This is a world made of students' answer to a question: what do you expect of a synthetic biology course like this? You can look at that. So the course went really, really well. We had engineers, Yanis and Slavik, giving us lots of practice in building logic gates and how electricity works in general. Then we had computer simulations and computer assembly of circuits, bacterial circuits that we then realized in real bacteria. And students were very happy about this course. I was very happy about this course because not only we managed to gather all these people interested in the idea of having synthetic biology community ready, it also allowed me to test the parts and the protocols that we were developing with NCBE in a real-world situation. Okay? And this event, this course that we run, was, I think, was very, very important for what's happening now. This was a critical mass of people and ideas that let the students then take it over. And this was like an initial push, and then when students, the alumni of the course, what they did, they set up a synthetic biology society at the university. They organized themselves to start a team. And now what's happening, we are in the second week of another synthetic biology course that I'm just running for them as an IG bootcamp to try Bryk assembly because we got the parts. We are trying to develop our idea for the iGEM project and so on. So we have 30 students plus few advisors working as an iGEM team this year for the first time in Rettberg. And we have other members of staff now asking when are we going to organize another synthetic biology course for our students. And now we're thinking about developing synthetic biology course as a practical activity for students that they can take instead of a research placement. during summer. So all these things start suddenly. If you have it done, if you get it done, lots of people come and want to use it, and the community grows. This is the single most important idea I can give you about synthetic organizing an iGEM. We started in February/March because we had some bureaucratic troubles in setting up synthetic biology society at the university. Once it started, we organized a brainstorm meeting and we discovered that the major source of funding, the BPSRC fellowships, we just passed the deadline. Okay. So this is the timeline that you can find on the iGEM.org website. And they give you a rough overview of an iGEM year. And this is my modification of it. So November you have the Jamboree. You go back from the Jamboree, you should immediately start selecting the next year's team. Ideally, I would think, by end of December, you should have your team selected. And then by the end of March, you should have your project selected. Because what happens later, there is a big break in April, and there are exams in end of May and June. So no one does any work anyway. For this chunk of this time period. So if you have your team here, and you can do it easily, I think, riding on the success of the previous year's team, and then if they run your recruiters for the next year, and you develop a project by end of March, I think that gives you enough time to gather your resources and to research your project before you actually go to the lab and do the work. I think Richard will tell you a little bit about this part of the procedure. So that is important to start early, and we are sure to start early this year for the next year. And then, of course, is the question of money. I know that at some universities, or essentially university pays for everything iGEM team needs. In our case, we need to find the money ourselves. And there are various ways you can look for the money. And for us, the first line of attack was approach the funding for iGEM as a funding for teaching. And we had a very nice case where one of the members of staff who wasn't invited to one of our iGEM meetings, he came, Rob Jackson from microbiology, came in, he just sat in the back with his arms crossed, he listened to us talking, and then he said that, oh, I know this guy, you should talk to him about getting money for your development of teaching resources. Okay? So we wrote an email to Judy and Bryk, who told us to apply for a Teaching and Learning Development Fund that the university runs for exactly this sort of thing, development novel approaches to teaching and engaging students in research work. So we wrote the grant, and a few weeks later we had the first £5,000 on our account. This was the money that we used to register, to buy the parts, to buy reagents necessary for our project. With having this money in hand, we went to the systems engineering people and asked, oh look, they gave us £5 grand. Will you give us £5 grand as well? And they gave us. So we now are halfway through a funding year, because we estimate that roughly for a 10 to 12 person team, we need about £20,000 to go to January this year. And so it goes. Now having 10,000, we are now pushing the School of Biological Sciences to give us another 5,000, and we applied for another grant to the intra-university annual fund, which funds an unusual initiative throughout the university, and if we're lucky, we might just get it. And that's, of course, in addition to all the sponsorships deals or crowdfunding deals that we are considering. Okay. And going back to this connection between engineers and biologists, you should, I think, Richard may correct me, I think the really cool projects are the ones that really engage both sides of the community, so engineers and biologists. Because in our case, it was very interesting because I felt it was the engineers who were better prepared to argue for their biological project than the biologists were for their biological project. So that's something in biology that they found interesting and fascinating that they want to push for it. And they did. So we have a little poster in the poster session, and what we'll be doing this year is building a solar-powered fuel cell that will use cyanobacteria to generate electricity that we will harness to power whatever device the engineers will come up with. Okay, so this is cyanobacteria that we'll be working with, Synaptocistis, 683, most likely. And this is the fuel cell that we built using yeast, just normal microbial fuel cells that we built using yeast during the course, as NCBE sells these fuel cells to schools. So this is solution of yeast with methane blue as mediator, donating the electrons to the electrode and powering a little motor that is built encased in metal, so that we know it works with electricity. So if you want, when I think is over there, raise your hands, people. Raise your hands, people. You can go in touch with me about the project and things that we'll be working on. And here we are. And I'd like to thank you for your attention now. Thank all these people who were instrumental in setting this all up. I think we have pushed this train fast enough so that now it goes up by itself. Jan, Sławek, Yoshi, engineers, Rob, Sheila, microbiologist, Lion, bioinformatician, Julian Park, director of teaching, John Scholar, my bosses from NCBE, and of course all the students who made this process. Thank you very much.
Well, thanks, Jarek. And please remember to pin your questions until the end of the talk so that you can move the panel. We'll move right on to our next speaker. So, today I'm happy that so many iGEM teams are here with us today. But what makes a group of people a team? And do you have what it takes to succeed? One man who definitely has answers to these questions is Dr. Richard Kelwig. Next to his research, he has always had an entrepreneurial drive. Early projects included the founding of the Enterprise and Engagement Club at the University of East Anglia, and entering and becoming one of the finalists at 4 business competitions over the years. Most recently, he has been the founder, scientific advisor, and project manager of 3 successful iGEM teams ranging from 2011 to 2013. And this culminated in his advising role through the Imperial team of 2013, which obtained 3rd place at the World Championship at MIT, gold medal, and Best Biochemistry. So today, Richard will share with us his knowledge and experience and get you inspired through the next competition. So please give a warm welcome to Dr. Richard Kelwig.
I'd like to say thank you very much for a very complimentary introduction, but I'd like to say that all of the activities that I've been involved with has very much been a team effort, and as I'll argue and discuss during my presentation, really the advisors of the iGEM team are the guys at the back. So this is the advisor perspective. We're sat watching you guys the star of the show, and we're just there to cheer you on and to give you that kind of momentum to help you do it. So really, you know, I'm the guy at the back pushing you guys forward. So I'd like to talk today from a little bit from an advisor's perspective, but I'll move on through the talk to talk about some of the big lessons that I think we're learning in the 3 iGEM projects that kind of I've been involved with, and what we all learned together, what the team during those programs. So, as Laura mentioned, I've been an advisor to 3 successful iGEM teams now. First of all, I helped set up those 2 iGEM teams on the Norwich Research Park, East of England, and this was with Dr. Richard Goldwater, who I think is in this room somewhere, that will join us later. And we actually set that up, first of all, in collaboration with the John Innes Centre and colleagues across the Norwich Research Park, and it was a really great year. I won't go into too much detail about the project, because I'd like to talk about some of the lessons overall, but essentially we worked with Martin and Aldi with the new chassis for technology, but also iGEM. and we arranged the UK meetup that year as well. So we know just how stressful it can be to organise these things. And then in 2012, I was advisor to the Norwich Research Park UVA Norwich team. Again, led those across the Norwich Research Park. We built some of the first components of a living tattoo, and I'll talk about that in a bit more detail later on. We also organised the UK team meetup again, and this was actually held at Google Campus, and so we used kind of the live streaming technologies that we're using here today. So pretty exciting to see that UK teams have carried on that tradition of developing forward. And we were awarded a gold medal that year as well, so really great project, really exciting. And then of course last year I was an advisor for the Imperial College's iGEM team in plasticity, where we essentially turned non-recyclable waste into a bioplastic. It was a really great year for us. We came third in Europe and then at the World Finals at the World Championship in MIT. And it's great for you guys this year, especially the new teams like Redding, where you can get straight to the kind of the World Championship and really enjoy that kind of global jamboree. It'll be really exciting and you'll really enjoy that, so it will be great. And then hopefully as well, ACS will develop next year, which they developed last year, which is to invite all the iGEM teams to basically write a peer review publication. And so we've submitted ours, and hopefully that that will be published later in the year. So it's a great opportunity. So I'm pushing ACS to keep that up, keep it playing out. So as I mentioned, I'm really talking from an advisor's perspective. And you see, this is where we are at back. Myself here, colleague Dr. Alex Webb. It was really bright, kind of sunny day, and that's why we're just awestruck of kind of iGEM. It's so bright and shiny. It's really a great day. And then Professor Paul Fremont and Professor Richard, who are co-directors for the Centre for Biology Innovation, for the assembly side. So we have a lot of great support from Imperial, of course. These are the guys that I'm going to kind of talk about the rest that we with last year. So if we're not the star of the show, what is our role? Well, I've tried to kind of summarize it as three main things we can do. First one is to provide kind of core resources. So you heard a little bit about that. And when you start up an iGEM, even if it's something that the institution does regularly, there's still a lot of resources which has to be gathered. And you guys are great at finding the majority of the resources. So it's by no means just the advisor's role. We just get a little bit to begin with the registration fee, and hopefully through like the Biochemistry Trust or other sources, hopefully some side you guys. But you guys have to go out there, talk to the companies, get in-kind support, and bring all the exciting stuff. Hopefully we can help you to begin with. This second part, inspire world-class ideas. As advisors, we really want to push you guys to come up with great project ideas. And if I'm honest, this is actually the easiest part of the whole job, because you guys come up with far better ideas than the advocate. As advisors, we're always telling you why you can't do something, and then you actually go and do it wrong. So that's not the difficult part, and so don't be afraid to push with good ideas. And then the third point is, I think as advisors, it's really important that we instill ideas about what it is to be a professional scientist or a professional engineer, and really instill ideas about responsible innovation. iGEM is great because it's really embedded within the culture of the project, helping you to think about great ideas, but to achieve them in a responsible way. Something which iGEM calls, which I'll it's actually quite challenging being an advisor because, as you heard, often we have to break all the rules of the institution and help set up all these different activities, break down the barriers in the institution and bring you guys together. But really, it's not us really doing that. It's the barriers come down because they see the enthusiasm and excitement from you guys, and that's really what achieves it. And so for us, the reward comes when we see you guys getting excited about your project. In the Jamboree, we've been really excited about that, which is really where we get our reward from as advisors. It's also great to see the dedication as well. So you can see here, this is a virus LD. It was optimized for bacteria with bioplastic last year, and you can see it actually had an infection. So that's what you think of. So basically, science doesn't always work. Sometimes the experiments are really difficult. Things just don't work. But you guys persevere. You're so dedicated to your project. You carry on, and and seeing that in you guys is fantastic. So don't give up, keep trying, you will break through, and it's great and advice to see that. And of course, we all get to celebrate not just the achievements of our own students that we've advised, but all of you guys, all of us in the community get to do that at the Jamboree. So we're all excited for everyone at the Jamboree, which is great. And because I've been an advisor to 3 teams now, I've seen a little bit of how a lot of the team members actually, iGEM has a very positive influence on it, in that direction. So many students go on to masters, courses, PhDs, to start companies, to do a full range of different things. So it's great to think that iGEM is a positive kind of role in kind of future direction. And so as an advisor, it's really great to know that. And of course, many of you will become advisors for future teams as well, which is something you should hold. So during iGEM, all of us, the advisors and students, we all learned a lot of stuff. So we learned about ourselves, our strengths, our weaknesses, what we're good at, what we enjoy, what we're passionate about. And that's great. But really what I'd like to do is just stay focused on kind of one important lesson that I think I learned and the teams learned overall, and give them kind of overall perspective on kind of one core important lesson, which I think will be valuable to each of you as well. So to start off with the first iGEM team that I was advising and started with others. And really it was all about introducing a kind of photosynthetic organisms. See the team here at the Jamboree. And so this was UAJC, and we wanted to introduce the chassis here, this box, and also this technology as well. So we were really focused on introducing these 2 new chassis and developing the technologies that would enable other iGEM teams to use these chassis. And it was a really great project. One thing we didn't have a full chance to explore because it's such an intense process and you have to do quite a lot in such a short period of time, is that we didn't get to fully explore the applications for these chassis. So we built, a lot of the technologies to help us use them, but we didn't realize its full potential as, say, a chassis for producing, say, biofuel or a material or whatever you want to do. So it was very technology-led, and that can be very powerful. But what I'd like to talk to is how you also need to have a great concept for a great application. Because synthetic biology is all about building stuff. So it's a great project with a little bit of technology-led. The second project, so this was in 2012, with NLP UEA Norwich Team in 2012. And we came up with this great concept, which was a living tattoo. It's absolutely phenomenal. So the idea is you had immortalized cells embedded in your arm, and you'll have to fluoresce different colours to give you indications of health. So it's a living thing, it's a part of you. And this was really great. It's not to talk to people, not only about synthetic biology, but how would you feel about these technologies becoming a part of yourself, and really kind of explore that. So it's a very clear and very strong concept, and it's absolutely fantastic. And concept-led projects can be a fantastic idea, especially in iGEM. It's a place to have very big ideas, and certainly if you have a very big idea, you can engage the public quite a lot as well. And as part of the project, we built 4 working biosensors, so we've got some really great technology as well. But again, within the summer, we couldn't build a living tattoo and embed that in someone's arm for lots of reasons. So it was very concept-led, and we built some working technology for biosensors to do the whole thing. And so when you go to the Jamboree, you will see these 2 extremes, and then you will also see teams where they try and balance between the 2, where they have a great concept with some great technology that more or less realizes that technology. And so I think that's the challenge, especially now that iGEM competitions have matured quite a lot. It's great. And so my kind of experience with plasticity is that the thing which drives the balance between these 2 extremes, having this great idea, having this great technology, that's just kind of cool in itself. To balance that off and have a better product is human practices. So just to give an overview of some of the logic that happened through the competition. At the start of the competition, basically the students came to myself and the advisor and said, we have this big idea, and that waste is a big issue. And they showed us this image, and it is. This is some of the waste just from Europe alone. And if we were to pile up all the waste generated over the last several years, it would effectively compete with Mount Everest. It's a huge amount of waste. It's an incredible challenge. It's a big thing. And so the students said, well, why don't we degrade this plastic? It's still pretty big. It's as big as one of the oldest mountains in Europe, and it will help the world, change the world, this idea. And it's mostly made of these oil-derived plastic kind of waste, like plastic bottles, things like that, food packaged waste. So we thought, okay, that's a reasonable idea. We could have just gone straight ahead to the lab. But instead, we said, well, why don't you go and speak to something about it? So we went to waste management companies, and they said, that's a terrible idea. Because actually, if you bring that plastic mountain to us, we can recycle it, and we can make new products. What we need to do is go after this special class of waste, this solid recovered fuel, which is known in industry, which is a mixture of waste, plastics, fiberboard, and paper. And we can't do anything with this. We just have to burn it or landfill it. So this is the real mountain. Still a mountain, but it's a slightly different mountain to what we thought. Spoke to some more people, and they said that waste is a resource. So not only did we choose the wrong mountain of waste, but we're trying to destroy it when actually we should be using it as a resource. So again, that changed the project very, very substantially, but very powerful. The next idea, people said, well, actually, focusing on the oil-derived plastics a little bit too much, but actually, bioplastics have a bright future. So why don't you think about those as well? Big market for bioplastics growing. Other people said that waste management is not just technological, it's not just when you talk technology, it's social as well. Maybe we don't create certain kinds of waste, maybe we encourage more recycling. That's part of the solution as well. So don't just be technology, like think about the social aspect. So all these ideas are kind of swirling around in the students' heads during the project development. And this is a kind of a brainstorming session that the students did to really kind of bring all those ideas together, and that's how the final project idea emerged, which is engineering microbes to convert rubbish or plastic rubbish into bioplastics, or as it was refined in the Jamboree. Essentially, we want some SRF, and then non-recyclable waste, into a bioplastic to make into products, or trash to treasure, as the students and thinking responsibly as well. I want to talk about recycling that new kind of plastic that was being made as well, so that we're not creating another mountain for the next iGEM team to think about. And then the technology came from that perspective. So the idea was refined, and the technology was designed around that idea. So I won't go through all this detail, but effectively, technology was all about having to target that non-recyclable waste and then make it bioplastic. And the refined that. And so when thinking about your project ideas, don't just think about the big idea, do that, this big ideas. And don't just think about a cool little bit of technology. Think about how you can blend them together. And the best tool you have to help you balance that is human capital, which really means going out there into the world and talking to lots of people about how you do that. And to finish off, I'd like to thank Dr. Richard Goldwater, Professor Judy Armitage, of Biological Sciences, and many of the colleagues that I can't list that helped to start up the first iGEM teams. And then, of course, your college, Professor Paul Friedman, and Professor Richard Kidney, the Kelwig Centre. Again, great supporters of the Centre. Many of the colleagues that have helped, of course, and the reason why I always mention Bob is that because the true stars of the show, just the iGEM students, which always make iGEM such a great community to be a part of. Thank you very much.
Thank you, Richard. Now for our keynote speaker. So we've talked about the education, we've talked about the competition, and surely we could go on to the ethics, the economics and politics, the health issues that can be tackled by, and even the art that can be produced through synthetic biology. I can honestly say, and I know I'm not the only one, that synthetic biology will become a big part of all of our future lectures. For a scientific field so young, it has tremendous potential, and even though it has a short past, our final speaker will show you why synthetic biology will have a long and amazing future. Originally starting out as an engineer and spending more than 30 years in the computer industry, luckily for us, he set his sights on biology these past years, becoming a principal research engineer in MIT's Department for Biological Engineering from 2005 to 2011. As part of this pioneering synthetic biology, iGEM spun out of MIT in 2012 and became the independent non-profit organization we know today behind the premier student synthetic biology competition. So last, but definitely not least, please give a warm welcome to the president of iGEM, Rettberg.
Okay, well, thank you very much, and thank you for participating in iGEM. So, how many teams do we have? So, what team are you on? UCL. UCL, okay. Someone who's not on UCL? Norwich, okay. Imperial, UCL Norwich. Ready?
Cambridge.
Cambridge?
Kent.
Kent? York?
Sheffield.
Which one?
Sheffield.
Sheffield? Cork? Cork?
Leeds.
Leeds? And where else? Exeter.
Imperial. Manchester.
Imperial? Wow, that's like a dozen. Did I get everybody?
Warwick.
Warwick? Okay, Edinburgh? Wow, that's great, that's great. So I do not have slides this time. Sometimes it's hard to coordinate what I want to say with the slides. That's why you'll see when I give presentations, I often just show pictures of the students. Then I can say anything. So I'd like to tell you a little bit about iGEM's history and kind of where we are today, what iGEM is about. And then I'm going to tell you a bit about synthetic biology and what happened with that and what's going to happen with that. My goal in this presentation is to expand the range of your thoughts a little bit, to help you avoid preconceived notions that might be less useful, but to give you an idea of the kind of things you can do with the rest of your career. Okay? Because it's really, there is a big, long future available. iGEM started out at MIT. Tom Knight had started doing synthetic biology before I got there. I got there in 2001. We had many lunches. So we would buy lunches and people would show up. So that was really the start of synthetic biology in iGEM. In 2002, we thought, well, there's January where MIT has no classes. What are we going to do? So we decided we would have a little design class. And so we did that in January 2003. We had 16 students. We set up the pattern for iGEM at that point. We told the students that they would design cells that would blink. And that sounds like a fine plan. We said we would have the DNA synthesized and they'd be able to test the cells in the summer. That was like way optimistic. But we had a good time. And we did it again in the January class of 2004. We also got some money and we ran a very quiet iGEM competition, 2005, in the summer with only 5 teams, and they were all friends. So if it didn't work out, we wouldn't tell anyone. Okay? So that's the first lesson was you have to take a lot of risks, but you have to be willing to have some failure. So we tried to do that. We continued on with 2005, and since then we've been growing and growing. We've been international all the time. So this year we have our official number of teams, 245, and that's roughly balanced between Asia, North America, and Europe. And there's about a dozen, 15 teams in Mexico and South America. In previous years, we had regional jamborees. And last year we had a jamboree in Hong Kong, Toronto, and Santiago, Chile. on the first weekend of October, and Lyon, France, the second weekend of October. Out of that, only about a third of the teams were able to come to the MIT Jamboree. We exceed the size of MIT. We can't do that. Can't have everybody come. That had a problem, which is it meant that some teams couldn't participate all the way to the end. And there was a little bit of crying. We were not very disappointed with some teams. And this time we said, okay, we're going to do it. We're going to do the giant jamboree. So all the teams are coming to Boston. We have the Heinz Convention Center, which is right across the river from MIT. And it will hold all the people. We have lots of rooms, and everybody can do all the presentations. It's a little bit longer, 1 day longer than it has been in the past, so there's enough room for all that. And this time in particular, we're working harder on making it so there's time for the students from the different teams to get to meet and know each other a little bit better. Because you really are going to find that there are people who are in Japan, or Korea, or Canada, or Chile, who've been having the same problems you've been having all summer long. And their cells had to transform, and all the same problems as you. And that is great. And as you go forward in your career, those people will be your friends. So this is an important thing, and we're making extra room for that. We've also added some tracks this year. We have a track in community labs. This is because the DIY bio community is starting to get its act together. And there are now organizations that have safety committees that actually have insurance. They become serious, and we want to include them in our community. Along with that, we have had a problem in the past of young students and older students. One year, we had a 35-year-old giving the presentation for his team. The 18-year-olds thought that might not be really fair. In retrospect, the 18-year-olds did a better job than the 35-year-old, but so be it. So we developed this undergraduate and overgraduate sections. It also lets us give out more awards. So that's cool. The general opinion from the judges last year was the projects in the undergrad sections were better than the project in the overgrad section. We'll see what happens this year. That allowed us, by the way, to do the community labs. I don't think about it as excluding, but rather it was a way for us to open iGEM up so that people of any age could participate as overgrads. We also put in a track for art and design. And so we'll have artists providing entertaining and stimulating views of what synthetic biology can be about. And we have designers. If you think about the world of Apple right now, you see design kind of in some ways at its peak. The products that are coming out are all products you want. You want them because they're really cool and they're really they solve things in a way that feels right. That's because of excellent design. Some of the things that have happened with Daisy Ginsburg, for example, and some of the teams have shown you what that design is like. So we're having a track specifically for that. We have a software track, and the reason for that is that yes, teams do software projects, but programmers are unique. They don't always work in the lab. They stay very late at night programming, right? And so we have space for that. Just like that, we have a track for policy and practice. And the policy and practice track is for people who are going to go and join the government, are going to work in think tanks, are going to figure out what the policies of dealing with new technologies ought to be. People who will be economists. You don't really have to be able to pay that. You have to be able to write. So we have these two tracks, we have a few other tracks. In addition to those tracks, vertically, like the other normal tracks, we also have some awards. So you could have a policy practice team, but somebody else could win the policy practice, human practices award in iGEM. You could have a software team, or software could be a part of one of your teams, and you could win the software award. In addition to all of these awards, there aren't so many. We have too many teams to give everybody awards. We also have medals. And the medals are very important. Everybody can win a gold medal if they try and meet all the requirements. For all the details, go to the website, and the judges are sorting that all out, and they'll have all the medal requirements and the requirements for each track all set appraisal. So all of that is happening. We have had our high school division going along at the same time. We have 50 high schools this year. They are having their jamboree at the end of June. We'll be flying back to attend that. We have 50 high schools. About 40 of them will participate in the jamboree. That's still small enough that it fits at MIT, but we're trying to figure out what to do. High schools are different primarily because the constraints on a high school student and a high school team are much tighter, more severe. For example, they mostly don't get to work during the summer. Their schools are closed, their teachers are off doing something else, and perhaps their family is on vacation. So we have done that as an experiment. Very interested to find out how that works. So the main thing to say about iGEM is everybody come to the giant jamboree. It's going to be great fun. You'll meet everybody else, and it'll really be the right thing. Everybody come. I'd like to say a little bit about the values that we're teaching in iGEM. When I started this up, there were a couple things I did. The first thing was I talked to the biologists at MIT, biology professors. They're smart people. And they told me unanimously, Randy, this is a bad idea. They said, undergraduates, they don't know anything. They can't do any synthetic biology. And the summer's so short. It turned out they were just wrong. We did it anyway. So as you were saying, you have to go and do it anyway. Don't listen to that. And the second thing was, I talked to Woody Flowers, who's the person that started the robotics competition. I don't know if you're familiar with that in here. It's very big in the US, 2,000 high school teams. And I asked him at the end, I said, so what question didn't I ask him? And he said, you didn't ask what values you teach. And I said, that's right. So let me tell you what the values are that we care about. There's the first three are the effort teams put in. We appreciate the very hard work. Occasionally we have a team who works very, very hard, but their technology doesn't work. And then they think they shouldn't come to the jamboree. Please come to the jamboree anyway. We value the effort that went in. And you can share some of the lessons that you learned with everyone. Number two, we value the accomplishment. The teams all over do amazing things. It's a problem. If you think about the problem we have, we do iGEM, and then we want to tell people what the projects were. We say, it's on the website. Watch the videos. Set aside the next 2 weeks. It's going to take you that long. So there are too many good projects coming through. So we value them. And some of the projects spark. And you'll see that at the jamboree. You'll see that in the website. You say, oh my goodness, what an amazing project. How they do all that. And that's wonderful to do. Along the way, we have values that are core values of being a responsible individual. The first one is respect. iGEM is a community where people are trying very hard. They deserve your respect. And if you're feeling like things aren't quite fair or anything like that, share that with us. We have a complaints email. But remember that everybody else is like you. And they're all trying and working and doing their best. The second one is cooperation. I'm going to come to that one at the end of my talk. The third one is integrity. And this is something where I'm going to put it real clear. Integrity, from my point of view, means intentionally telling the truth. And a funny thing to say, right? Because you lie or you tell people no. It's going out of your way to say what is truthful. If you are going to be scientists or engineers, remember that you can't fool Mother Nature, right? The scientists have to figure out how to tell the truth. It's kind of the core aspect of being a scientist is the scientific method, the peer review, all those things are about finding the truth even when your own prejudices lead you in a different direction. So in iGEM, tell the truth. Intentionally tell the truth. Don't think, well, maybe I'll hide that. No, tell the truth. It's good for you. It's good for everyone. So when I come to Europe, I'm often asked for the definition of synthetic biology. Because let me define synthetic biology. Or let me tell you a story. So let me tell you who I am. So when I was 12 years old, I decided that I wanted to design computers. I happened to live in Champaign, Illinois, or Champaign-Urbana in the University of Illinois, and they'd been designing some big computers. And so I got excited, and I wanted to design, and I knew that's what I wanted to do. As I went through the rest of school, everything I did was about how to design computers. And I read the books, the IBM Principle of Operations that told how the computers worked, learned about all that, studied like mad. When I got to MIT, I took the introductory class in every field. So I figured they'd teach me 80%, and that would help me design my computers. After that, I went to back to the University of Illinois, avoided the Vietnam War, and studied physics. I thought that'd be important for my designing computers. That turned out to be very hard, but not necessarily such a good idea, but I did it. When I graduated, I went, along the way, when I was young, I knew that designing computers meant I would work for IBM, because only big companies could do that. That's equivalent today of saying, well, if you're going to graduate and you're going to get a job in industry, it's going to be with one of the big pharma companies, right? Because that's where all the jobs are. They're the only people that are doing anything. When I actually graduated, that wasn't true. It turns out that there were lots of companies. The mini-computer had happened. And that mini-computer revolution meant that suddenly you could design a computer. In fact, I designed the computer we use in the ARPANET as the second packet switch, me and a friend. Now, I was a little weird, he did it too, but we designed it, and that was because there were these ETL integrated circuits. So, I'm an engineer. I'm not a scientist. I have friends that are scientists, but I'm an engineer. I want to make things that are used. That's my goal. I don't particularly want to write papers. I've written a few papers, but mostly I want to make things. So the job that I got first was with a company called Beranek and Newman. It was to join the group that did the ARPANET. The ARPANET was the first of the packet switching networks. Joined in 1972, which I was around in 1970 as the beginning of the network. My job was to do the link to Europe. So I started to come to the UK. There's a satellite link that connected a very experimental network in Europe to the ARPANET in the US. That satellite link turned into the technology of Ethernet. So that worked out pretty well. Along the way, I got to write the first TCP/IP for Unix. I got to design the computer we use as the packet switch in the ARPANET. And then I designed a machine which is a parallel computer that we shipped in 1981 to be 256 processors. It was about, oh, not so big, from here to that wall of racks. So all of that was designing computers. They weren't used very much. It was also doing all the networking. So I was at the beginning of the internet. I didn't invent the internet. I did write the first TCP/IP for Unix. But I got to be there and watch, and I know the people who did. So from that, I draw analogies of what will happen in synthetic biology. And I can explain it a little bit. After being at PBN, we formed a company to build computers, parallel computers. They kicked me out like Steve Jobs. And then I went to Apple, when Steve Jobs was not there. And there I got to actually be on the team that developed one of the Macintoshes. And so there's an engineering experience. It was like being in a room with about 40 people and knowing without any doubt that everybody in that room wanted to make that computer exist. They didn't have a second goal. They wanted to make that computer exist. They wanted their name to be on the inside of the case. And they wanted to be able to tell their friends 40 years later, I designed that. And then many, many people want it. That's the pleasure that an engineer has. Now, I'm kind of focusing on engineering. I'll come back to some of the reasons why that's happening. But actually, engineering and science go together. If you think about the computer revolution, and you think about even the programming revolution, that actually helped the science. So material science, surface quantum mechanics, a lot of money flowed in to the science because of the importance to it in the computer industry. The reason that we got to these very high density circuits and very complex designs was that it was a constant back and forth between the science of materials in particular and then the needs in the computer industry. So I think the same thing happens in synthetic biology. I think that synthetic biology is, to me, mostly engineering, but there's a strong tie with the scientists and the biologists. So some of you will often become engineers and you want to make these things. You want to make the trash to treasure real or other ideas. And some of you will want to be academics and will want to run a lab and an academic research institute. Mostly what you see around you, of course, is the academic research environment. So I'd like to say a little bit about why synthetic biology is based in the engineering environment. But let me give you kind of a big view first. So let me tell you about engineering. So engineering is kind of a funny thing. If you say electrical engineering, is that a part of physics? Right? It should be. Or computer engineering, software, that's part of math. What makes it different? What makes it different is primarily that in an engineering discipline, you're trying to make something intentional. So, for example, you might be trying to build. You could build with stones, right? You go around here and you find buildings made out of stones. Here in Oxford, there's a lot of buildings made out of bricks. What's the difference? It's not that a scientist went out and found a stone that was rectangular in that shape and had holes in it and said, we need more of these, go find more of those. It was an engineer who said, we need a stone that's a consistent size. And we could have had holes in it so it was lighter, and so concrete and mortar could go in. That principle of making the devices that you want so that you can build the things you need is a core part of engineering. We say they're abstractions. That's the main thing that engineering produces. You develop an abstraction. I think scientists actually do that as well. But engineers make an abstraction, and then they design things to meet that abstraction. That's not happening in synthetic biology very much right now. People are mostly going out and they're finding natural things. They're kind of cutting up pieces of DNA. But these abstractions can be physical. It can be this piece of DNA, you assemble this piece of DNA because you abstracted it into a part. Another abstraction that's very important is a model. You could say, this is a model. It is not actually correct. It's an abstraction of the real model. Now, if you think about it for a second, that's what happened in electronics. Right? Physics solved the problems. Maxwell's equations solve the problem of electronics, except you can't solve them. So instead, you develop the idea of inductance, capacitance, resistance, which are related to Maxwell's equations, but they are something you can use on a regular basis. But each of those is an abstraction that's valid for that particular component, only over some range of operation. A resistor will change its resistance with temperature. Inductance in a resistor will have a big effect on the actual performance of that resistor when you ignore it at low frequencies. Okay? So we are right now lacking a similar abstraction in how biological systems operate, in an abstraction that would let you do easy computation. One thing that happens in an electronics device is you will have a power supply. The power supply will be regulated. And regardless of what the load is, the voltage will stay the same. That's very, very common. It's been going on for decades. Same thing doesn't really happen in a cell. We're not using cells that have a power supply that is regulated, that holds the energy molecule concentrations, the ribosome concentrations, and all the concentrations constant. Everything interacts with everything else. When we get to a point of really engineering, we may have to do those things to ourselves. We may have to make them behave better, make them be less like stones and more like bricks, designed for our intentions. So those are things that come with standardization. It also comes from the idea of designing, not craftsmanship. So you're designing your device so that it will work the way you want, rather than here's something I'll just push on it a little bit. I think those are things that are big and important in synthetic biology. Now, along the way with that, we're going to have to make catalogs, tools. We started off by making the iGEM, it used to be the Registry of Standard Biological Parts. It's now the iGEM Registry of Standard Biological Parts. How should you think of that? The first is that it's got a nice collection of parts, and by having teams use those parts and make new parts, we have a community. So you can use the parts that other people made, and you can take your parts, whatever you're making, and make it available to everybody else in the future. That's an important part of standardization that happens in engineering. Imagine that you are designing something for a computer. Maybe you're making a graphics board for a computer. Okay, you're really smart, make a great graphics board. If you don't make it so it plugs into the computer, nobody buys your board. Right? Doesn't do you any good to have something even more brilliant if it doesn't plug in where it's supposed to plug in. The idea of parts in the registry is that they are designed so that they plug in where they're supposed to plug in. It's part of your work as an engineer. So when we think about the iGEM registry, we don't think it's the only registry. We think it's the best, but we don't think it's the only one. Because to think that it's the only one would be to be living in some kind of a pre-Copernican world, where we're the center of the world, center of the universe. No, there should be a lot of other registries. Other people will have points of view. Other people will have a specialty. Perhaps they'll specialize in some plants, or maybe they'll specialize in some particular kind of who knows what. When you go to stores, you don't find just one store. You find a lot of variety, and you enjoy that variety. You wouldn't expect that all of the grocery stores in town would all publish in one catalog run by the biggest store. No, everybody has to have their own points. Same thing with the registries. There will be a bunch of them. And after there's a lot of them and a flourishing of lots of different registries, there'll then be a consolidation down to a smaller number after we understand. So the iGEM registry is important now. I think it might continue to be important over the long run, certainly be important 5 years from now. So I'd like to say a few things about quantum. The first is the iGEM registry is a funny piece of software. It is a wiki, the media wiki engine. And so when you go to the pages, you edit them like you edit a wiki. That turns out to be a wonderful thing. Wikis are just great. They are a real website that everybody can modify. And you don't have to have a Unix account. Also, it keeps track of who made what modifications. So if you made a bad modification, then somebody else can go and remove it. And they can say, no, it's a good modification, you can add it back in. So all of those things, and they're all being tried out by Wikipedia, and with quite a lot of success, we have adopted into iGEM for your registry pages and your project pages. The second thing that happens because it's a wiki is, if we thought about a normal model of a catalog of parts, we would say, well, here are the parameters that a part should have. Here's perhaps it's a promoter. Here's how strong the promoter is. If it's a inducible promoter, then, well, it's induced by this molecule, and here's how sensitive it is. But what if it's a part that is buoyant, that makes gas vesicles that make your cell float? Who would have ever thought to add a column for buoyancy? How buoyant is this thing? And if you thought of that one, how about if it's supposed to produce an antibiotic resistance? So we have to have columns for, the number of columns expands way too big. So the problem isn't a computer problem, it's that we don't know what we want to be specified. It's like you go into a grocery store and they say, oh, well, we have sugar, we have milk. There's no variety. That'd be bad at a grocery store, right? You love all the variety. And so we want to allow the variety. The way we allow that variety is by having the registry at its core be a wiki. But at the same time, there's a real MySQL database there. And the MySQL database is, in fact, separate from the wiki. And it stores the sequence with parameters, categorization. If it's a composite part, it stores how you specified it, and how all that is broken up into the basic parts. So if you have a full real database, And you can get the information from that if you're a computer in XML. So people who are writing software can write perfectly fine software, get the data out of the database in a way that software understands. At the same time that people can get the information out the way they understand. Because of the wiki, the registry in recent years has developed a large number of highly documented parts. I won't say whether they're well documented, I won't comment on the quality of the measurement. But many parts from iGEM teams over the past few years now have a lot of measurements, a lot of photographs, microphotographs of the cells, of colonies. That is now the new standard. A comment to all of the iGEMers: when you look through the registry, you will find some parts that are badly documented. They'll be like, here's a part that looks pretty good. It says, this is the part I used. And you'll say, that's a stupid part. And you feel bad. But look at the name of the person who put it in. Okay? That's the person who put it in at that level of quality. And then remember, as you put your parts in, the next person's going to come along. And if your parts aren't well documented, they're going to say, oh, look at your part. Oh, yeah, she didn't do a good job. Right? So do a good job, because the people who come after you will be looking. Okay? So that's about iGEM and the registry. Time. Okay? We are talking about the field of synthetic biology and what happens. Time. The title of the talk is Short History and Long Future. Let's talk about time. So I have a question. What Oxford? Is Oxford older than science? Anybody from Oxford? It might be. When was Newton? Well, you know, before, you know, kind of, I think around 1100. That's about when Oxford, right? I think that a lot of science was kind of the form. Somebody thought about things and figured this must be how it is. And people tend to think, well, this is how the gods must have made the world. And that was kind of the deal, right? And if you couldn't figure out anything, that was probably because the gods were, you know, causing you troubles, right? So things take time. The next comment is, we use a lot of analogies when we describe synthetic biology, but analogies fail. Why do they fail? The first reason is, I tell you an analogy from my youth about punch cards and floppy disks, and it makes no sense because we don't have the shared background. The next way it fails is that the analogy wasn't really right. Trying to tell an analogy in order to make some point, but A, it's not really B. So I'll be using some analogies. You use them because you're trying to say something a little too complicated to be believed or understood. So I'm going to talk a little bit about what has happened in the past, because I think it shows why that can happen in the future. So synthetic biology is about 15 years old. These days I round. So about 2000, it's about 2015, about 15 years old. Remember that the computer information revolution is now about 50 years old. About 7 years. We're kind of 1940 to 2010, about 70 years old. Go back. Think about the computer world about 15 years in, sometime around 1955. You can't do that, right? You weren't there. But I remember the capacitors, and you actually could open them up and unwind them, and they had aluminum foil in them. The resistors were really pieces of carbon with lead stuck on the end. And some of the resistors were wound around like wire, like that. Looks like an inductor, because they were inductors. I remember making a radio out of a piece of pencil lead and a razor from a shaver, and headphones, and listening to radio coming from Chicago, 150 miles away, with a setup like that. The radio in here doesn't have any of those parts, right? Their software does the radio in here. But this is decades later. So now let's do a transformation. Let's think about synthetic biology 15 years in. Think about it as like 1955 electronics. There's no digital electronics really, little bit. The computers of those days were good at arithmetic. And you wouldn't need many computers because how much addition do you have to do? Because the world needs a lot of addition. Right? That's what they're good for. People said that, right? How much addition does my computer do? My computer is not calculating spreadsheets. My computer is spending a vast amount of computing horsepower listening to the cell signals. That's what it's doing. And when it's not doing that, it's showing me movies. You'll never imagine that, right? So if we think about electronics now, it's now highly manufactured. It's been going on for decades. Hundreds of millions of dollars at least have gone into it. And we're just not there. But we will go there. So if we think about the long future, my experience with the internet, and it's kind of a funny thing, right? You get to be a part of the internet from early days on now the early days of synthetic biology. How lucky, right? So I remember giving a talk around 1997. And I was on the internet, and I was talking to this collection of the audience, and saying we've been having about 80% growth for about 27 years. If that were a financial fund, would you invest? Obviously, you would say, well, it's been going like that for 27 years. It must be ending soon, right? And what I told the audience was, instead of having that thought, have a different thought. Imagine that it continues. And say, well, if it continues, what must happen? What will the world be like? When I was at Sun Microsystems, after I left BBM, I joined Apple, and then for 4 years, and I was at Sun Microsystems for 6 years. I was the Chief Technology Officer of the Storage Division at the end of it. And so a lot of very good. But I said, in those meetings, they said, you know, the thing that's going to really go is phones. That's going to be the computer in a while. Their phone was that they thought that would happen soon. It took quite a lot longer than they imagined, probably an extra 15 years. But it did happen. That is a normal problem when you do the extrapolation. So in the early days of the internet, the phrase was the internet changes everything. That's too simple. Where's the modifications? Everything except this is a mess. But I think people are now have pretty much come to realize the internet changes everything. It's like the Industrial Revolution now. The Industrial Revolution changed all of society, right? It changed corporations. It changed everything. So we had the Industrial Revolution. That was about energy. The tool that you used to deal with energy was the machine. We've been going on that for, what, 150 years or so? Are we done? Probably not. We don't have little micro motors everywhere, although we're getting closer. Then we did the Information Revolution. I think of that kind of 1940 on. We're clearly, at best, halfway through the great impact of that. We're talking about the Internet of Things right now, for example, and we don't have Internet everywhere. And I can't have wireless everywhere. Right? We're getting, but you can imagine that when you actually really are connected all the time, and everything really is connected all the time, it's a different world. Very, very different world. You may not notice it, but the number of people who are going around like this all day long is going to be really big. And they bump into things, right? This is a huge social change. I don't know quite what they're doing, but there must be somebody else at the other end of it, right? So that idea of the internet changes everything. Now we have synthetic biology. What's synthetic biology? It's a revolution too. The technology revolution clearly has medical things. The one that matters is matter. What do cells do? Cells manipulate matter better than anything else. Cells put an atom where they want the atom to be. They put a molecule together in exactly the right way to make a little motor for a vagina. Right? It's gorgeous. Number one, we know that it works because it exists. We don't quite know how to do it, but we're making progress. And we know how to do some things. That's why this, as a revolution, has validity. Think about some that didn't work out, right? I made a little list. So where is fusion? Fusion power. We know the sun does that, but we're not doing so well. Let alone the personal fusion packs for your house or your car, right? So Back to the Future, where it has the fusion, who knows, right? Or another one, flying cars. You know, I was driving up here from London. I wanted a flying car. This would be much better, right? Anti-gravity. God, I saw that in all the videos. Anti-gravity is like, where is it? World peace. We didn't do so well on that. We spent the first half of the 20th century trying. We didn't do so well on world peace. So, take synthetic biology and switch things around. Right now it's new. Imagine that it works. Suppose that it's like the internet. We get better and better and better at it, and we keep on getting better and better and better. You know Moore's law with number of transistors doubles every year? What happens when, I don't know, maybe it'll be Laura's law. It'll be synthetic biology, blah, 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 every year. And then you can start to predict it. And you realize that all those things about being good at matter are happening. What's the world? Right? So the question is going to be, remember this long future. So in 50 years, what will your grandchildren want from synthetic biology? Can you give them world peace? The some parts are going to be really interesting, right? There's wealth, poverty, power. Those will still exist. What will they be like in a world where we're good at matter? Right? Prejudice, pride, status, society. Those things will all still exist. But in a world where the technologies exist to do matter, what's going to happen? So, as we develop synthetic biology, we have two general paths. One is, who's going to be the Microsoft of synthetic biology, or the Apple? Or, what about the startups in Africa, in Latin America, all over the world? Will synthetic biology be something where one answer meets all needs? Or will it be something where there's a need in every corner of the world that can be met with synthetic biology? We're placing that bet with iGEM. That's why there are teams all over. That's why we care so much that there are teams all over. It's why we try to push it out and democratize it into the DIY community, high schools, to everybody. If you think about programming, don't think about computers, not industrial experience. Think about programming. The idea of there's one piece of software we need to solve each application, that's gone away. Apple's App Store showed that there's a need for millions of applications. We can keep having programmers forever, all over the world. Never run out of the need. Maybe synthetic biology is like that. If we do that, that will help world peace. Because it won't be centralized. It won't be something that somebody can take power in. So I'd like to end, and I know I might have a little time problem, but my last page was something I went over in our values. Cooperation. Cooperation in community. That's a funny thing to talk about when what I have is synthetic biology competition. Right? But the normal thing that happens in science, and it happens a fair amount in business and engineering, is secrecy. Keep things private. You keep things secret. Keep your work hidden so that nobody scoops you. Right? So if you're doing some work in the science, you do your work secretly in the lab, everything's secret, you don't get scooped. Right? If you are working at Apple, you keep it very, 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 very secret because there are a million people trying to find out what Apple's going to release so they can get a little bit of a lead. But I'm sorry about the fact that science has gone in that church. I think that secrecy and worry about getting scooped in science is a very, very big problem. In iGEM in the future, we'll probably try to work on that a little bit. Because it's not the idea that you're a little smarter than I am, I'm a little smarter than you are. Bill Joy has a law. His law says that no matter where you are, even if you're in Oxford, there are more smart people outside your organization than inside. Right? So however smart you are, there's like one guy, right? One woman, who's the smartest. Everybody else is somebody smarter. Right? So if you keep your work secret, then what's going to happen is nobody will tell you what you got wrong. And you will continue to do that wrong thing, and continue to do it, and continue to do it. And you'll just be wrong. Also, if you keep it secret, you won't find out that you have a lot of friends who'd like to work with you on it. So what we do in iGEM is we provide an environment where there's competition and there's still sharing. We think that's the world of the future. We look at what's happened in social media, in Facebook, where there's an openness there never was before. And we think that there's a lot of future there. If you see things, there's a book called The Zero Sum Economy or Zero Sum Society, which is talking about this idea of the community resources as being the key. And we, Tom Knight, Randy and I, Jerry Sussman, have pushed a lot in the direction of open science. That the information should be shared because everything will happen faster. Everything will happen better. And there's the third thing, if you keep secrets. You'll make enemies. You'll feed jealousies. Open's better. We think open's going to work more. So that's what iGEM is all about. In iGEM, you will learn all of these things. You'll learn about teamwork. You'll learn to suffer and struggle, stay up night, eat a lot of pizza. You'll learn that the experiments don't work. You'll get mad at some of your teammates, and then you get happy with your teammates. Last year, I had been told that iGEM was breaking up relationships. So I went and checked this out. So I go around the teams. Okay, so now how many relationships were broke up with this one? And actually, there was a little bit. Actually, these are university kids. They might be. This might happen anyway. And then I started to hear about relationships that were created in iGEM. And I felt much better. So all of those things will happen. The competition and the giant jamboree will be just the most fun ever. And then you'll be done. You'll write your papers, get published in ACS, and you'll go on to the rest of your life. And for all the students and the instructors and advisors, that's what matters. You'll have learned how to share, you'll learn how to compete, you'll have learned talents that were never going to be, you'll never have experienced in your undergraduate life otherwise. You'll learn how to fundraise, right? So all of that will be there. So thank you very much for participating in iGEM, and I hope you get all of those values out of it.
I just want to move on to the question and answering panel at the top. We'll put some chairs in front, and then you can ask away. So just give us a couple of minutes, or actually 1 minute.
Okay.
Sorry. Yeah. You have the same with those.
Great. So, I have your attention, please. Does anyone have questions to start with? Okay, so I think if I'm right, we've got a question from Yale, from Sven Wilteres.
Yeah, could you speak up this one?
Yes, but what's the question?
The question is to all three of you. What is the most memorable iGEM project you have seen?
Tough one.
That is actually very, very difficult. Like you said, there's so many great projects, and they're all very diverse. They're never in the same area. Of course, the projects that I've worked with, some of the ones that I remember the most. But some of the projects that really stand out to me, and the reason why I kind of spoke about how to kind of balance kind of the idea against technology, that I think now as the field moves forward, and I said very eloquently, it's all about building stuff. So those projects where teams actually build things really stand out, I think, now to me, and really, I think, show how the field is maturing and moving forward. So teams that build fuel cells, for instance, teams that have built sensors to detect spoilage meat, a great team from 2012. Those kind of projects are the ones that really kind of stand out to me, the ones where I could trace everything go.
I went through a couple of projects to show my students what iGEM can do, iGEM teams can do. And I really like the UCL Plastic Republic thing from 2012, I think. It's an idea of aggregate plastic that pollutes ocean, making a giant island. And how they approached the crowdfunding for it is they sold land on this island. And they got like 1,500 quid from it. And they managed to weigh bacteria, detect and aggregate pieces of plastic in water. And there was some clever engineering involved. And I think in this project, along with the human practices, they also went out and did some biobrick assembly with the public. Really tied out everything together. They had all the elements really nicely done. So I, as an educator in education, I use this as an example of a very sort of complete iGEM project.
I am pretty careful to not have favorites, and certainly not during the year. The thing that strikes me is also at the jamborees I often have to meet and greet and don't get to see the presentation so much, but there are projects that didn't make it into the finals that are quite excellent. I remember that there was a collection of projects that were essentially about bacteria in cold water. There was things to produce an antifreeze so bacteria would freeze. This was at the time of the oil spill in the Gulf of Mexico, where the water was warm and bacteria was happy. And some of the reason, perhaps, why that wasn't as bad as it might have been, the bacteria ate up oil. We had an Exxon Valdez spill up in the Arctic regions, where the water was not so nice. And if the bacteria had been made to be able to survive in that colder water function, that could have been very significant. I also like the ocean plastic island plan. I thought that was very clever. And that's another problem that might be useful. It also points to what you might do after that. You might take the cyanobacteria aspects to provide energy to do even better things on the island. You might use the floating gas vesicles from the Rettberg project, which is always one of my favorite projects, to help control where your island is, or maybe even to make your island sink if that's what you needed to do in the future. So To me, I like the broad range of projects. There are ones that just start. I remember one of the years when Cambridge had made chroma proteins. I listened to the project before that. I said, oh my goodness, nobody can ever do that. Now it seems the best project ever. And then the next one's better. So I think that what I like is the depth. I'll bet that I thought about writing a book. It was a compilation of the top 20% of that. And something which would only, you'd have to do that, so you would have something which is only 700 pages thick, so people would read it or not. It goes pretty deep. I remember a project that had multiple colors that were localized to different parts of the cell in order to code a library. So just by looking at the cells under a microscope, you could figure out what the particular DNA was that had been put into that particular cell. And they actually had that work, and they didn't get it. I didn't even know if they advanced. So there are a lot of great projects. It may well be that we have a dozen, 15 teams here. So not everybody's going to be rocket scientists, but I'll bet the other projects are going to be great too.
Can I take a question from the audience? You're all baffled by the answers. Great. I actually have a question. So in terms of advancing synthetic biology and looking at education, would there be a certain level of age where you think we should focus on? Do you have to focus on A levels or the undergrads or the graduates, or you think maybe even the general public?
We certainly run material transformation with 10 years. We have worked with 43% like this. So you can do this type of practical. In terms of, I think it should be taught practically as early as possible. But there is a series of legal and health and safety regulations which have to cope with to introduce iGEM experience to high school. Right now, if European secondary schools want to participate in the high school edition of iGEM, they really have to be associated with a higher education institution that would take care of the health and safety and GM regulation too. But there are efforts in place to develop a subset of parts and protocols that would be allowed in a typical school without any regulatory backlash. So we definitely should start at least secondary school.
Absolutely, I don't think I can add much more to that. I think it's synthetic biology is something we should use, and iGEM as a whole does use to inspire people of all ages, all backgrounds, in different ways and different forms. And that's one of the great things about the competition, that in one sense, the next generation of trained scientists, engineers are moving forward, careers built to start to become scientists. In a sense, they're also inspiring society as well to think about and to imagine with the kind of what the history world will be like. As Randy said, we'll get better from these kinds of things and what that will be.
I do think the undergraduate is a good spot right now. It's clearly working, able to raise money. They're able to put concerted effort together over a summer. So that's working. The high school has exactly the problems you're saying. In addition, has the problem of the high school teachers often not really having a strong enough education to help the students very much. All these things we can work on changing, and I very much appreciate the idea of developing pieces of the technology that can be used there. Because there will be students who are, and at young ages, at 14, 15, 16, kind of beginning to set their careers. I don't know if it's necessary they be able to do it very deeply, but I believe it might be important for them to be able to do it something, and then to imagine what they're going to do as they get older. And that can be an inspiration for them to learn a lot more, to learn the things they need, and to say, well, I have to learn this science so that I can do that engineering in the future. But over the longer run, there is a big puzzle of how this becomes something that everybody makes use of, and how does it become something that everybody understands. In the early days of the computers were viewed as dangerous because they were giant brains, they were going to take over the world. Right? I used to joke with my friends that all the iGEM computers had a big red switch on the side, really big actually. I said, try to take over the world, put the switch. Right? But of course, we now know that computers did take over the world. Right? We have a little arm, the World Wide Web, right here in our pockets, right, tracking us, taking care of everything for us, and you can't turn it off. So it turned out it was okay. But that worry will be there. There's always been that worry. There is an extra element in synthetic biology, which is that we happen to, it's life, the topic of life, where we value life and we don't define life very carefully. It is completely okay to squash a mosquito, even though it's alive. Perfectly okay to put bleach on your bacteria and kill billions and billions of bacteria, flush them down the drain. But it's not quite the same to go and re-engineer your dog. It's okay to put GFP in a plant. All of these things are questions, and the piece of it is disturbing the natural environment. And the second one is that we are made out of the same thing those things are made out of. And is it okay to re-engineer something? How do we know we would re-engineer ourselves? Somebody might not try, or something might not happen by accident. So I think that in fact, it's been pretty safe compared to other industries. Certainly the number of deaths, illnesses compared to Industrial Revolution and coal mining has been much better than that. So perhaps as we get used to it, we'll kind of understand and accept better. And that will make it possible for more people to be able to think more directly. That is a core reason why you have to educate the public. But even more than that, it's going to be a great adventure. The information age, like the space age, are wonderful adventures, and the public should participate with joy.
On that note, we have another question.
Yeah, so this one's from me. Yeah, my name is Avi Roy, and I'm a biomedical scientist. And first of all, thank you, all three of you, for a wonderful talk. So I work on aging, and I also work on lab-grown meat. And I look at synthetic biology as a superset of techniques, but in the category of techniques that can be applied to solve problems, just like genetic manipulation is. So it kind of rolls onto what you guys were talking about, especially you, Randy. How do we, I'm speaking from the perspective of Europe and other parts of the world, not US or Brazil or Australia. How do we stop synthetic biology from becoming the taboo that GM has become? So, I mean, and GM doesn't directly apply to humans, but it still is taboo, at least in some parts of the world.
Success, good examples.
Well, GM has success in the US, great.
No, but I think that GM has some good examples and some bad examples. It hasn't been dealt with very openly. You have the Roundup, the engineering of crops so that they are immune to Roundup, so you can kill the weeds. You have the predatory behavior of some of the large agribusinesses. So a little bit of what's happened is that's wrapped up in the corporatization that's happened in the United States in particular. And so if you live in a world where large corporations get to do as they wish, then you may fear they don't do something in your benefit. And then you have to figure out about what is dangerous. I think that is possible. I think that as we have more iGEM students and more schools that are participating, they get to see it closer up. And they see the possibilities that are really good. And it may be that what happens is that the public has been through a period where other stresses caused them to worry about everything. And perhaps if that can get fixed, they don't have to worry about all of our things quite as much. If that can't be fixed, then other problems come. And that's synthetic biology doesn't have too much to do with that.
I'm hopeful for synthetic biology not to recapitulate genetic modification, because I think the level of community involvement, both from the synthetic biologists themselves and both from governmental organizations, I think is better or much more deeper and broader than it ever was back in the 90s, as far as I know. And I think that the community that iGEM is creating through the also safety regulations and human practices that it imposes on teams and continues to impose and maybe strengthen them in one way or the other is a big part of that. Because we should remember that this is the beginning of the road, and we're nowhere near as advanced to cause any real world damage using synthetic biology right now. And yet, with the synthetic biology dialogues, we have Jarek Institute publishing its own assessment of security within synthetic biology. We have gene synthesis companies voluntarily imposing screening against pathogens and other dangerous sequences on the orders they receive. We all have that already, and I think this is a great. There is hope. I hope that this will continue to develop like that, so that we can avoid any catastrophe.
Absolutely. I would just kind of add to that just a little bit that this whole event is streamed live on the internet. People have direct access to the discussions that are ongoing, and all of you, I'm sure, and of course myself, have been to many meetings, events, public engagement activities, a whole mix of different things where you get people from policy positions mixed together with artists, designers, members of the public, scientists, engineers, all together. It's very multidisciplinary. So lots of people come together with different ideas to shape in a very open way what's happening. And the field is very self-conscious about a lot of those kind of issues. It's prepared to ask, should we be doing a particular thing? Should this application be something we consider? Or should we only think about other applications which ultimately maximize the public good of the field and the technology that we're building? I think that's why synthetic biology is different, because it's built from the ground up, and through iGEM we're training everyone to have those kind of questions at all levels throughout the entire development of the projects, not just at the start, but throughout as well, and to shape the work that they're doing to maximize.
Hi, my name is Edward Robin from Genetics. We make software for synthetic biology. I want to give our software to all the iGEM teams. Please come find me afterwards. What are the challenges involved with running a registry? So you've got information problems, physical DNA problems, and what are the best practices that you guys have found in order to get around these issues?
So the major problem in running a registry is contributions to it. Why do people provide you things? If you were running a registry for a company, for a company that was offering as their product DNA pieces, parts, then you'd understand and say, oh, well, our marketing department is in charge of that. The engineering group and the product planning, they have run different sections of our catalog. And then the catalog's purpose would be to get the customers to buy. There are industrial catalogs where there's a fair amount of information about the things yourself. Normally, what you would have, we use the example of nuts and bolts commonly as interchangeable parts. But if you think about it, those are so standardized that they're intended for a nut that I buy from this company to work for the bolt I buy from this company. And that has been worked out over decades and decades. And therefore, in a registry of that sort, you would simply refer to the specification number by some complicated number. And then you'd say, and this is a 632 volt. And you'd be done. And then you'd say, oh, and it's made out of this material, it has this kind of a head, wherever you do. So the core problem in running the registry right now is that it's very unclear, it's not worked out. What is the right information, parameterized information to store? As I said, it's easy if it's a promoter, you think. But suppose it's a coding region for a protein. Well, at least you'd like to know what fraction of the time a protein is actually successfully made from the mRNA. You definitely need to know the mRNA lifetime, because the extent to which it degrades is going to cut down the amount of protein generated. You need to know that the protein degrades. You need to know in that process how much of all the different molecules in the cell are used, and what concentrations are necessary for it to perform whatever function. There's a lot of information about every part. And I think it's quite impossible at this stage to be able to say, oh yeah, every part should have these 5 things. Even though people are saying the core problem in the registry right now is parts aren't measured, I think that is incorrect viewpoint. I think it's incorrect because, number one, we don't really know what to measure outside some very simple things. Even if we were to measure them, we don't know what environment to measure them in, so we would get it right. And finally, actually, a lot of the new parts are measured in their application, and there are lots of curves, lots of words, and lots of pictures about parts that have that method. Beyond that, the next problem is that the contents of the registry, because they're created by students, sometimes aren't right. But this is also true in Wikipedia. So what we have found recently, we spent a lot of effort on this. What we found recently is that there are a lot of parts that have more information, where other teams have come up and made use of a part, these measures, and they've added that to the registry. In our registry right now, if you look at a part, you'll see experience, and now there'll be a little green number. That number is a logarithmic kind of representation of how much documentation is on that page. So now you'll go to some part and you'll see this little green 5. That means there's a lot more there. You should click on that and go check it out. The entire question of the quality of the DNA is partly a result of teams sending us the DNA at the end of the year. We are desperately hoping for cheap DNA synthesis soon, or a larger amount of money spent on DNA synthesis. My hope would be that within a couple of years, teams will not send us DNA. They will specify who will synthesize them. And we'll get rid of all those tubes flying all over the world, and we'll just synthesize. And then, a few years later, we won't send it out anymore because everybody will synthesize. And at that point, the registry becomes the information. We all want that. But, you know, we have wanted that for quite a while. In fact, about 2009, there were strong arguments that we should stop sending out distribution because everybody should just synthesize. And it was only $1.50 base. That was wrong. We will accept DNA and send out DNA until synthesis is just obviously better. And it starts to look silly to do that. We'll just keep on doing that. It's very efficient, right? But the other part that's interesting about registries is that, I think it's right here, is your company going to be the center of the world? Which company is? It's going to be Autodesk. It is. Is it going to be one of the companies that's starting up? Someone who's very successful? When you, as a synthetic biologist, go to work in the market, whose company's software are you going to log into? In the old world, that was a normal kind of question. Of course, you log into Excel. If you were a financial person of any kind whatsoever. You're just like yourself. You live in there forever. And if you were an artist who's involved in images, you'd probably in Photoshop. And Photoshop worked on making more plugins and links and fancy links to the other software so they could be the one world. I think that your will perhaps be technology if there never is one tool. And if instead a whole web of interconnected tools and viewers and repositories of information and labs with their own data comes into existence. That's what I'm trying for, hoping for. That's one of the reasons why teams that make tools for iGEM can plop their tool right on the page. It's one of the reasons why we're working towards providing hosting for tools from iGEM teams so that those tools can keep existing at the team level. The common problem with iGEM teams is that your team will have a programmer, the programmer will write code, and the very best he keeps under his website will be wonderful under his bed. When he graduates, he's going to get a new computer, and the website goes away. All the people in the future lose the wonderful work you've done. That's why we insist on everything beyond our servers. It's no other. We just have had that happen in 2005, I believe. We lost more than half of the web pages from all of the teams because of exactly that. They went away. So the answer is to figure out how to be a part of a community of sorts. That means standards. It's almost certainly the case that standards we have today are not adequate. We have to figure out new ones. That would be very, very hard. Make contributions to those ones. That's a long answer. But the reason for that long answer is that people think that the way things are today, there's a common fault. They'll look at what you have done and published and say, I can do better than that. That's normally a good thing. But they forget that the work you published is the work you had done a year ago. You kept on working. You're actually maybe the expert. They have to be what you're going to publish in the future. In the registry, the people who are making new registries, they have to not keep the registry they remember from a few years ago, or the registry that exists. They have to keep the registry that's going to be built.
The other speakers want to comment on that?
I wish there was a standard where I could switch between different softwares, make protocol, and then I could have assembly starting in Snap and switching to Autodesk and finishing online using Photoshop. That would be cool.
Please take notes.
I think as well it relates to a broader issue in kind of science, and one of the reasons that I think synthetic biology is such powerful, because if you see those studies where 50% of the top scientific publications in biomedical science, people can't reproduce that data. There's a real reproducibility issue, and it does go down to a lot of contextual things. The system that that biological system was being examined, it's the conditions in which it was being examined, it's the way they measured that kind of behavior. It might be specific on four kind of say measure fluorescence in objects with units, and even the same model, but different machine will give you different values. All of those reasons mean that in biology, reproducibility of data is very difficult and very challenging. And so one of the core things in synthetic biology is to kind of not kind of just accept that as kind of a given, but actually to really look at what are we actually measuring, what is the important information we need to measure, and how is that useful. And so you'll have perhaps not only different repositories where we think of kind of static information, but people looking at different ways to measure the behavior and function of parts. And maybe you need to have systems where you can pull all that information together to help you model and forward design biological again, the key components of technology for design is behavior. So you need a whole range of pool of information to bring that in. And so when you look at part registries, no one person will be the guy who has all the answers and will have all the information for any particular part or pathway or system. And so you do need to have data to be able to move around and understand what that means. So it's diversity which is really valuable.
You know, in travel sites, there are sites in a number of different shopping malls. There are sites that don't actually sell you the product at all. They just go and check a lot of different sites and tell you what the best is and summarize. You can imagine there's things like that. It's kind of information about what a whole lot of different tools have to say about this part. But they don't have any information themselves at all. The other thing is that this year we have done a very funny thing, which is we sent out a measurement kit for all the teams to go and measure the same thing. And our intention is to bring all that data back and see just how much variation there is from lab to lab, and see whether maybe everybody is actually getting the same answer or not.
Well, thank you. Unfortunately, we've run out of time, so I'd like you all to give a thank you again to our speakers and panel members.